Hello, my name is María José Tárrega, and this work is part of my industrial PhD research conducted jointly at the Universitat Politécnica de Valencia y Global Omnium Medio Ambiente. In this study, we investigated how microbial communities respond when wastewater treatment plants begin co-digesting sewage sludge with pig manure. Using 16S RNA sequencing and advanced ecological analysis, we explored how the ecosystem adapts, which microorganisms drive this transition, and which operational strategies maximize the methane production. Co-digestion with pig manure represents an excellent opportunity to increase renewable energy production in wastewater treatment plants. However, it also presents a major biological challenge. High nitrogen concentration led to ammonia accumulation, which can inhibit the microorganisms responsible for methane production. In addition, it has traditionally been assumed that biomass requires long acclimatization periods before it can stability process of pig manure. Our work directly challenges this assumption. To investigate this phenomenon, we operated 6 mesophilic anaerobic digesters for 100 days. We compared previously acclimated biomass with conventional biomass that had no prior exposure with pig manure. We also evaluate 2 feeding strategies, split feeding 3 times per week and pulse feeding once per week. Throughout the experiment, we monitored process performance and performed methanogenic analysis on days 0, 13, and 19. Before analyzing the microbial community, it was important to verify whether the process remained stable. As shown here, cumulative methane production increased continuously throughout the experiment, even when ammonia concentration exceeded 2.5 grams per liter. In addition, we did not observe any signs of inhibition in stability indicators or in methane production. When comparing our results with inhibition thresholds reported in literature, we found that the digesters remained stable within range that are traditionally considered critical. This observation suggests that the microbial community was able to adapt rapidly to the new operation conditions. Our first major finding directly challenges the idea that biomass requires months of acclimatation. As shown by the beta diversity analysis, digesters inoculated with acclimated and non-acclimated biomass initially displayed clearly different microbial communities. However, these differences rapidly disappeared and the ecological trajectories converged towards similar community structures. The statistical analysis confirmed that inoculum origin had no significant effect on the final community composition. The same pattern was observed at the process level. After approximately 0.6 hydraulic retention times, methane production became nearly identical in both inoculum types. In other words, operational conditions were far more important than previous history of the biomass. From an industrial perspective, this suggests that a lengthy pre-acclimatization phase may not be necessary before starting a pig manure co-digestion. Once the inoculum effect disappeared, we investigated which factor was actually shaping the microbial community. The answer was the substrate. The introduction of pig manure triggered a profound ecological restructuring of the community. At the taxonomic level, we observed a strong enrichment of microorganisms belonging to the class Clostridia and group closely associated with the fermentative process in sintrophic metabolism. This shift allowed the system to maintain stable methane production despite elevated ammonia concentration. We also observed that disappearance of acetoclastic species such as methanosaeta and enrichment of hydrotrophic methanogens, including methanoculeus and methanosarcina. This taxonomic signature is consistent with a transition towards sintrophic acetate oxidation, allowing methane production to continue even under tan concentration, the 3 grams per liter. However, the most interesting results emerge when we examine microbial interactions. The pig manure fed community developed a much more connected integrated network than the control digesters. This indicates greater metabolic cooperation among species and a nascent ability to exchange intermediates such as hydrogen and acetate. In other words, the system did not adapt simply by changing which microorganisms were present, but by completely reorganizing how they interacted with the another. What was the impact of the feeding strategy? The weekly pulse feeding regime produced approximately 18% more methane than the split feeding strategy, with the same organic loading rate. From a methanogenic perspective, the pulse feed digesters exhibit lower Shannon diversity, indicating a highly specialized microbial community. Taxa such as Rhombusia or Turibacter and Methanosarcina were positively correlated with improved performance. The 7-day fasting period likely promote the depletion of metabolic intermediates, improving the thermodynamics of sintrophic pathways. Interestingly, several taxa identify as ecological hubs in the co-occurrence networks, including Methanosarcina, also show positive correlations with the methane production. This reinforces the idea that microbial network reorganization had direct functional consequences on the digester performance. Our conclusions were not based only in a single analytical approach, but on multiple complementary technicals pointing toward the same ecological interpretation. Taxonomic analysis based on data 2 revealed rapid convergence of pig manure-fed microbiomes. Diversity and coordination analysis confirmed that operational conditions were more important than inoculum origin. Network analysis revealed the more integrated and cooperative community under pig manure co-digestion. Finally, correlation analysis and Keystone taxa identification directly linked these ecological changes to methane production. However, all approaches support the same conclusion. The system rapidly reorganized into a more specialized and functionally efficient microbial ecosystem. To conclude, this study demonstrates that microbial adaptation during sludge and pig manure co-digestion is much faster than traditionally assumed. First, we observe rapid ecological convergence when operating conditions outweighed inoculum history. Second, pig manure did not destabilize the process. Instead, it promoted a profound sintrophic restructuring on the microbial community, resulting in denser, more connected, and potentially more resilient ecological networks. Third, feeding strategy emerged as a key operational driver. Pulse feeding increased methane production approximately 18% while promoting more specialized and functionality efficient microbial community. Finally, from an industrial perspective, our results suggest that prolonged pre-acclimatization may not be necessary when operational conditions are properly controlled. Overall, this work demonstrates that anaerobic digesters should not be viewed as a black boxes, but as a dynamic microbial ecosystem that can be understood, managed, and optimized. The microbial community did not resist the disturbance, it reorganized itself into a more cooperative, more specialized, and more efficient ecosystem. Thank you for your attention.